We'll continue with Dr. Patrick Brown from Johns Hopkins University.
Well, thank you very much, Erica. Thank you so much for that. It's a tough act to follow, but also an inspiration at the same time. So thank you to the organizers. I guess that's kind of thanking myself since I was on the committee, but Jeff really ran the show, so thank you. So a couple of things I just wanted to correct from your program is I don't have a PhD, so I crossed it out on my name tag. For those that have PhDs, I think it's important that those of us who don't recognize that we don't, because that's a whole lot of work to get a PhD that I didn't do. So I'm just an MD, I'm sorry. Second thing is I won't be talking about kinases, which my very first CCF grant was on kinases, but that was a long time ago. I'm going to be talking about what we're currently being funded in, and that's the epigenetic basis of relapse in childhood leukemia, what we're learning about biology and how we hope to target this therapeutically. So before jumping into epigenetics, I'd like to spend a little bit of time just making sure we're all on the same page for what we mean by epigenetics and how it relates to genetics. So at the risk of being overly simple, our cells have a nucleus in it. In our nucleus, because we're humans, there are 46 chromosomes, and those are in pairs, 23 each, come from mom and dad. And those chromosomes are organized by the rolling up of DNA. And in the DNA are contained our genes. So if we look at this a little bit more closely, chromosomes are packaging of a very, very long strand of DNA. The DNA is made up of a sequence of 4 different letters, which represent 4 different chemical components, and that is our genetic code. These are organized into genes. Genes at the DNA level are transcribed into RNA, and then that RNA is translated into proteins. And that's how the genetic code results in the proteins that function in a cell. Different proteins are expressed in different cells, but the same code is in every cell of our body. And it's the proteins that dictate how an eye cell sees, how a liver cell metabolizes, how a skin cell protects. It's the proteins, and not the DNA is the same in every cell, but the proteins are different. And that is explained by the regulation of which genes get expressed in a given cell. So problems can happen with this very simple genetic code, and those are called mutations. There's different ways to mutate the genetic code, but all of these result potentially in abnormal proteins that behave abnormally. Genetic mutations are the cause of many different types of diseases. Today we're talking about cancer. And cancer is, in fact, understood classically to be a genetic disease. And as we have become more sophisticated in the technologies that allow us to read the genetic code, we understand that most cancers, are genetically very, very messed up. That in an individual tumor, there can be hundreds, and in some cases thousands, when you look at melanoma and other types of solid tumors, of mutations within the cancer cells. So just to show some examples of hematologic malignancies, non-Hodgkin's lymphomas have about 100 mutations of different genes per case, per tumor. In CLL, it's about 20. In AML, it's about 10. In ALL, it's about 7 per case. And these are relatively low compared to cancers generally. But there are some diseases where genetics don't fully explain why the cell is behaving like a cancer cell. And probably the most striking example is infant MLL rearranged ALL, which is a disease I'll be talking about more, where there literally is only 1 mutation per case, and that is the rearrangement of the MLL gene. Genomically, other than that, the tumor is silent. And so we know that MLL by itself can't cause cancer, because if you put it into a mouse model, it doesn't cause cancer. There must be something else going on in the cell that takes that MLL rearrangement and then has it progress into a full-blown tumor. And the answer cannot be found by sequencing the tumors. And that means that some cancers, and maybe all cancers have a component of this, but some cancers may be uniquely epigenetic in how they arise. So what do I mean by epigenetics? Well, if genetics is the DNA sequence, then epigenetics is the code that determines how that sequence is organized so that a cell knows to either express or not express a gene. And the key component of that is contained in the red circle in the histone complexes. So histones are protein complexes that the DNA strand wraps around. And very importantly, there is a code, a epigenetic code associated with these histones that determines whether the DNA sequence in that DNA is open and able to be expressed into a full gene and protein, or closed and not being able to be expressed. So that's shown a little bit in more detail here. When a gene is contained in DNA that is wrapped tightly within histones, then that makes the DNA impossible to be read and transcribed into RNA and protein. When the histones around the gene unravel and open, that gene is now exposed to the machinery in the cell that transcribes into RNA and translates into protein. And that's controlled by changes in the chemistry of the histone proteins. There are post-translational, after the protein modifications, and typically these are acetylations and methylations of key amino acids on the histones that control whether the histone is open or closed. That, in conjunction with another element of the epigenetic code, which is methylation within the promoter sequence of the DNA. So shown here, there are so-called islands of C's followed by G's in the genetic code that exist in the promoters of genes. And those are the parts of the DNA that regulate whether a gene is being expressed or not. And when those are methylated, the CpGs, which is another modification, chemical modification of the C, when it's methylated, that's associated with genes being not transcribed or not expressed. And when they're unmethylated, that's associated with genes being expressed. And there is strong crosstalk between these two processes. When the histone code causes the gene to be wrapped up and not transcribed, the promoter is also methylated in most cases. When the histone code is causing the histone to be open and the gene open for expression, then the promoter tends to be unmethylated in the CpGs. There's strong crosstalk between these two. And together, this is what we think of as epigenetics. And so, cancer is a genetic disease, but there are certain cancers where the genetics really can't explain why the tumor has become such a malignant process. And that's where epigenetics may come into play. And so let's talk a little bit about this disease. So infant leukemia is defined as leukemia in an infant. And that's a pretty simple and straightforward definition. This baby's unimpressed with the logic behind that definition, but it is what it is. Infant leukemia is unique in that 80% of babies with leukemia will harbor a specific genetic abnormality, and that is a chromosomal translocation that involves a gene on 11q23 called MLL. Another unfortunately unique property of infants with leukemia is their strikingly poor prognosis with the best available standard therapies, including chemotherapy, bone marrow transplant. The cure rates are less than 50%. That is in stark distinction to leukemias in older children that can be cured in over 80% of cases. What is most interesting to me biologically is that when infants with leukemia relapse, it is after they have already achieved a very deep remission, and yet the relapse happens very quickly. Most high-risk forms of leukemia, when the patient relapses, it's typically associated with a suboptimal response initially also. That if they have minimal residual disease at the end of induction, then that will identify a patient group that's high risk. Infants don't behave like that. The actual remission rates with initial therapy is very high. So their disease starts out being very sensitive to chemotherapy, and yet they relapse within a very short period of time. And when they relapse, their disease is now strikingly chemo-resistant. It is nearly impossible to put these babies back into remission. And so that really suggests that over a very rapid time course, that there has been emergence and selection of a chemo-resistant population in a way that's unique to this disease compared to other types of cancer. So this just shows the outcomes of these babies. So the 80% that harbor these MLL rearrangements have cure rates in the 40% range, whereas infants that don't have MLL rearrangements, which tend to be older infants closer to a year, have outcomes just as good as older children with ALL. So it's really this MLL rearranged subgroup that has this unique biology and unique clinical presentation. So I hope I've convinced you that new treatment approaches are badly needed for this subset. And the kind of poster child for novel therapies that change the outcome of a leukemia is Ph positive or Philadelphia chromosome positive ALL, where with the incorporation of a targeted agent, a tyrosine kinase inhibitor, Gleevec or dasatinib or others can do the same thing, the cure rates went from being among the lowest in pediatric leukemia to now being on par with other forms of childhood leukemia, even without transplant. And so our hope is that there may be novel targeted therapies that could improve outcomes in infant leukemia in the same way. And so why might an MLL rearranged leukemia be an epigenetically driven disease? Well, it turns out that the function of the normal MLL gene is an epigenetic function. So in the normal MLL gene shown on the top, the set domain, which is on the very end of the protein, its job is it's a histone methyltransferase. So it transfers to the histones the methylation group that tells it to open up and transcribe that gene. MLL does that to a number of target genes within the cell. The MLL gene was discovered as a key gene in embryogenesis. It is a master regulator of which cells turn on these genes and which cells turn off these genes. And it does it by controlling chromatin, by controlling epigenetics. When MLL is involved in leukemia, what happens is that N-terminal histone methyltransferase is lost, and what is gained is a fusion partner that has a different chromatin modifying enzyme called DOT1L. And that methylates a different histone protein. What that means is that you take the part of the MLL gene that binds to its normal transcription factor targets on DNA, and you link it with a histone modifier that is far different in what it does. And that what it ends up doing is amplifying all of the genes that normally are under MLL control, but amplify them until they're out of control. It also, through another mechanism, turns off genes even more strikingly than they should be downregulated. And so you get this global change in gene regulation within the cell after this fusion occurs. And so it makes perfect sense that this fusion may exert its cancer-causing properties through chromatin and epigenetic changes and not through the DNA sequence. And that's why we can't find any mutations in these cells when we sequence them. All right, so I'm going to skip over a whole lot of work done in our labs and others that have led to the following model of how infant MLL rearranged ALL occurs and why we think it's a genetic disease. Back in 2002, Scott Armstrong and Stan Korsmeyer's lab published what I think remains one of the seminal papers that looked at global gene expression. And it did it using an old technology, which was an Affymetrix array. At the time, it was the absolute cutting edge of technology. It allowed 30,000 genes to be examined all at once. And what stuck out in that study was that these MLL rearranged leukemias have a strikingly abnormal gene expression program. And what is true is that there are a set of upregulated oncogenes. Again, these are not mutated, they're just upregulated through epigenetic mechanisms, and that these genes, things like the Hox family, MEIS, FLT3, are activated by the activating histone code that comes with that fusion partner, that DOT1L. There are also a set of strikingly downregulated tumor suppressor genes, and together, this is what's causing this cancer to behave so badly, that there are oncogenes that are overexpressed, there are tumor suppressor genes that are underexpressed, and these together cause the cell to function abnormally. And yet, none of these problems would be evident if you just looked at the DNA code. So it is not a genetic disease, it's an epigenetic disease. There have also been, to support this idea, various epigenetic modifying proteins that are required for MLL to cause leukemia. I've mentioned DOT1L, which is the protein that's in a complex that comes along with a partner. There are also so-called reader proteins. These are proteins required to take the histone code and turn it into whether a gene is turned on or turned off. And the best example of that is a bromodomain protein called BRD4. All right, so our hypothesis then is that epigenetic changes drive relapse in this disease. So why might epigenetics be an important driver of relapse? I've talked so far only about how the epigenetic might cause the leukemia to begin with. Now I'm going to shift gears and talk about why I think epigenetics is really the explanation for why those babies start out being so chemosensitive, but end up within a very short time being so chemoresistant. So we know that CpG methylation drives the expression of genes. And so I've already shown this. Hypomethylated CpG islands are associated with the gene being expressed, hypermethylated with the gene not being expressed. Now, it turns out that every time a cell divides, the chances of there being a change in a methylation state are orders of magnitude greater than the chances of a DNA mutation developing. And both of those things can affect gene expression. So it makes sense then that if something is randomly changing hundreds or thousands of times more frequently, then that is a fertile ground for the selection of chemoresistant cells within a cell population. So if every time a tumor cell divides, its methylation state is going to change or has a risk of changing, then that's a very fertile ground for the selection of cells whose gene expression drives them to have a greater survival. And so we think this is a very potent substrate for the natural selection of chemoresistant clones. So the technique we use to get at the epigenetics of these cells and of these subclones is called bisulfite sequencing. So just as you can sequence the DNA code, the T's and C's and A's and G's, you can also sequence the methylation of CpG islands. And the way that's done is you first treat the DNA with bisulfite. Bisulfite will convert the unmethylated CpGs into uracil, but the methylated CpGs are protected from conversion. And so every time there should be a C and there's a U, you know that that's an unmethylated CpG. Every time there should be a C and there's a C, that's a methylated CpG. And that's bisulfite sequencing. It's a very simple technique. What that allows you to do is basically, on top of the normal DNA sequence, to sequence the CpGs as either methylated or unmethylated. Methylated is shown here as a 1 or a filled-in black lollipop. Unmethylated is shown here as a 0 or an unfilled-in lollipop. And so what we've defined as a haplotype, we take 4 contiguous CpGs, and that's the basic unit of analysis when we're comparing samples for CpG methylation. All right, so we've done a pilot study where we've taken 2 infants, both of whom were diagnosed with lots and lots of blasts in their bone marrow, both of whom went into a deep MRD negative remission within a month, and both of whom relapsed within a relatively short period of time. We chose 2 cases that have the same exact MLL rearrangement to take that variable out of the equation, and we performed whole genome bisulfite and RNA sequencing to look at gene expression so we could correlate those 2. So I won't go through the details, but our whole genome bisulfite sequencing resulted in full coverage of the genome to good deep levels. We defined each 4 CpGs as what we called an epiallele and looked at the percent methylation and the frequency of epialleles. And this is what that looks like. So this is an example of 4 contiguous CpGs, and you see 3 different reads of the same part of the genome. So in this very simplified case, the epiallele frequency of the fully methylated allele is 33%. The epiallele frequency of the unmethylated is 33%, and the frequency of the partially hemimethylated allele is 33%. And the total percent methylation of this entire epiallele is 50%. Half of the CpGs are methylated and half are not. Again, very simple conceptually. We also did RNA-seq, where we got gene expression for every gene of the genome at the same time. The first thing we wanted to know is, does CpG methylation correlate with expression within these patient samples? And in fact, they do. So if you look at the CpGs and the epialleles that are unmethylated, they're highly expressed. If you look at those that are highly methylated, there's a bimodal frequency. About half of the genes are expressed and about half of the genes are silenced. So we focus here on these genes whose silencing is associated with hypermethylation. We know that there are genes whose expression doesn't depend on methylation. Not every gene is under tight methylation control. And so this identifies a subset of genes that we're particularly interested in. The first thing we did was to look at recurrent. Yeah, almost done. What time is it? Yeah, I was supposed to go until 3:55, right? Okay. I must have misread the schedule. At any rate, the first thing we did was looked at recurrent methylation expression changes at relapse. So here we're comparing relapse to diagnosis. And what we found was that the key genes, this is an unsupervised look at the genes, that 3 genes that are incredibly important in glucocorticoid resistance, a key component of ALL therapy, came out as being important in this analysis. So what it tells us is that there's a tumor suppressor gene called LCK that when it's silenced has been associated with resistance to steroids, and that was strikingly downregulated at relapse. When we look at 2 other oncogenes that are associated with glucocorticoid resistance, NERP3 and PKM, they were strikingly upregulated, not because of mutations, which is what had been discovered in the past, but because of methylation changes. And this never would have been evident by straight sequencing. Another example is Ikaros. Ikaros is a tumor suppressor gene that has been identified by mutations to be really important in causing ALL. We have found that is epigenetically mutated or silenced at relapse in these 2 infant leukemia samples. So another way Ikaros may be involved in the development of tumors, the development of resistance, is through epigenetic dysregulation. And that's shown here. Red is methylated, blue is unmethylated. And that's associated with a 34-fold decrease in RNA expression by RNA-seq. So I hope this shows you the power of this analysis. So what we're able to do now is take every CPG island in every epithelial in the genome and show those that are whose methylation changes between diagnosis and relapse. So let me talk you through this graph. There are CPGs that don't change between diagnosis and relapse, where their frequency remains exactly the same. And those are so-called founder mutations that don't change over time. Then there are emergent methylation haplotypes. So these are fully methylated epialleles that are not present at diagnosis, but present in a high proportion of relapse. And then there are extinguished epialleles. These are epialleles that are methylated at diagnosis and are unmethylated at relapse. So we're particularly interested in this emergent. So these are epialleles that are unmethylated at diagnosis, methylated at relapse, and are associated with strong downregulation. That's where Ikaros comes in. It also suggests that if we incorporate demethylating agents into therapy early on, we may be able to prevent the emergence of these resistant clones at diagnosis. And in fact, when we look at the top genes in this emergent group, it turns out that 4 of them are identified as being mutated tumor suppressor genes that cause ALL. And so the top 30 unsupervised analysis, we found 4 bona fide ALL-causing genes that are emerging at relapse as being silenced epigenetically. Okay, so what this is leading us to do now is a further study where we're doing many more samples. We have grant funding from CCF, thank goodness, and V Foundation to do this work and extend on it. And we are, most importantly, doing a clinical trial that opened a week ago in infants, incorporating the demethylating agent azacitidine into their therapies so we can hopefully cure more patients. And then that's going to lead, we hope, to an international collaborative protocol with our partners in infant leukemia throughout the world. So we've developed novel analytic techniques to incorporate and integrate whole genome bisulfite sequencing and RNA-seq. We've discovered subclonal epigenetic changes that are either emerge or are driven extinct under the pressure of chemotherapy, supporting the hypothesis that chemoresistance may be driven by epigenetics, and raising the possibility that the incorporation of epigenetic therapies may be able to prevent relapse. We think this is going to be applicable in other leukemias and cancers as well, but we're starting with the disease where we think it's most likely to be of help. An incredible number of people have contributed to this work, not least of which is CCF, which has provided funding. So thank you very much.
there's a question there. So while the microphone is going there, what is the sensitivity of the method so that can you pick up if there's a subclone that forms 0.1%, that methylation pattern, was it there or is it acquired during the treatment?
It's a great question. So the sensitivity of the method. So the pilot study we've done went to 25x for each CpG, which doesn't give that kind of sensitivity where you could detect a 1 in 1,000 or 1 in 10,000 subclone. What we're doing in the follow-up study is identifying those parts of the genome which appear to be most important in driving this, and we're going to do reduced representation deep sequencing in those, and that will allow us to ask the question. What we suspect is just as in AML, for example, where you can, or CML, where you can find, when you use really deep sequencing, very small subclones of resistant cells that existed before therapy, and then are selected out through therapy. We suspect that the same is going to be true here, that those emergent epialleles that we talked about were probably there in very small numbers, and because they were chemo-resistant in very small numbers, they're the ones that grew out under the pressure of therapy. But it's a great question. It's going to require, to do whole genome bisulfite sequencing to that depth would be very difficult. But if we do reduced representation and focus in on areas like of Icaros and other things, then we'll be able to answer that question.
Pat, that was a great talk. Thank you. I had a quick question. You mentioned international study.
Yeah.
Is this mainly that you can enroll more quickly? And,
yes.
And what is your experience? Because I think...
So within, this is an extremely rare disease. Thankfully, infant leukemia doesn't happen all the time. We diagnose, there's about 150 infants with leukemia diagnosed in the US every year. That doesn't allow us to do very large randomized studies. However, if we join with the European and the Japanese groups, we can enhance the number of patients that enroll so that we can do randomized studies. What's currently going on is COG is piloting azacitidine. The European group, Interfont, is piloting the use of blinatumomab, a CD19 targeted immunotherapy in infants. If both of us show that these approaches are tolerable and at least have and are able to be tested, then that will be the randomized study between those two. We think we've gone beyond the point where we can ethically randomize babies to receive standard therapy. We know how bad it is. Unfortunately, our historical control is entirely too good. Every study done in the last 25 years has almost the same exact event-free survival, regardless of what is done. And so we have a very stable and unfortunate historical control, and it's time to do something better and different.